Okay, so let's just jump right into it. Picture this. You've got a solid and you've got a liquid. How do you take those two things and create perfectly pure, beautiful salt crystals? I mean, how is that even done?
Well, that exact puzzle is at the very heart of a key experiment you've got to know, the AQA GCSE chemistry required practical for making a soluble salt. And in this explainer, we're going to walk through exactly how to get it right. So here's the plan. We'll kick things off with the mission, then we'll look at the core chemical reaction that makes this whole thing possible. After that, it's the step-by-step method. We'll get into how to purify your crystals, and then we'll wrap up with a little debrief on how to absolutely ace the exam questions on this practical. Right, first things first, what's our actual goal here? The mission, if you will, is to create a pure, dry sample of a soluble salt. The classic example you'll probably use is making copper sulfate, which is soluble, from an acid and an insoluble base, like copper oxide. Now, before we even think about touching a Bunsen burner, we need our blueprint for success. Honestly, understanding the chemistry behind what you're doing is absolutely crucial. It's what turns just following a recipe into actually being a scientist. And this entire experiment hangs on one single process. It's called neutralization. It's a pretty simple idea, but it's super powerful. You take an acid, you take a base, they react, and they basically cancel each other out, leaving you with two neutral things, a salt and plain old water. And here it is, the word equation you've got to lock into your memory. Acid plus an insoluble base gives you a salt plus water. This simple line is the recipe for everything we're about to do. All right, we've got the blueprint, so now it's time for the execution. Let's walk through the exact method, step by step, so you know how to pull this off safely and get the results you want. First up, you're going to gently heat your acid. This just gives the reaction a bit of a kickstart, helps it go faster. Then you start adding your insoluble base. But listen, the key is to do it a little bit at a time while stirring. Don't just dump it all in. You keep adding and stirring, until you see some of that solid just sitting at the bottom, not reacting anymore. That's your cue. That's the signal that the reaction's finished. So why do we do that? Why do we add so much base that there's some left over? Well, we call that adding it in excess. And this is so important. It guarantees that every last drop of the acid has been used up. See, if we had leftover acid, our final salt wouldn't be pure, and the whole mission would be a bust. That unreacted base is our insurance policy for purity. Okay, but this creates a new little problem, doesn't it? Your beaker now has the salt solution you want, but it's all mixed in with that leftover solid base. So how do you get them apart? The answer is a simple but brilliant technique called filtration. You just pour your whole mixture through some filter paper in a funnel. The solid base gets trapped in the paper because it's insoluble, but the salt solution, where the salt is all dissolved, goes straight through. And just like that, you're left with a perfectly pure salt solution, all ready for the final stage. And here it is, the payoff. We've done all the chemical legwork to make our pure salt solution. Now it's time to turn that liquid into the solid crystals we were after from the very beginning. So you've got two ways you could go here. You could just crank up the heat and boil off all the water really fast. That's quick, but you'll end up with tiny, kind of messy, impure crystals. The much better way, the scientist's way, is gentle crystallization. It takes longer, for sure, but it lets these large, pure, perfectly formed crystals grow. And to get that nice, gentle heat, we use a water bath or an electric heater. This gives you this really gentle, even temperature, so the solution doesn't boil like crazy. It's that control that's the secret to getting those amazing high-quality crystals that are going to score you top marks. So the final steps are pretty straightforward. You gently heat the solution to let about half the water evaporate. This makes the solution more concentrated. Then you just kill the heat and leave it alone to cool down nice and slow. As it cools, you'll see the crystals start to form. It's actually pretty cool to watch. Then you just filter out your crystals one last time and leave them to dry. Mission accomplished. Okay, lab coats off, thinking caps on, time for a quick debrief. Knowing the method is one thing, but knowing what the examiners are actually looking for, that's the real game changer. So in the exam, they're assessing these specific skills, called apparatus and techniques. AT2 and 4, that's your filtration and your crystallization. AT3 and 6 are all about handling the acid and the other chemicals safely. See, every single step we just walked through is tied directly to one of these points. And are also testing your working scientifically skills.
This is all about the why. Why did you add the base in excess? Why did you use a water bath instead of just a roaring Bunsen? Answering those questions shows you're applying knowledge and evaluating methods. That's WS 2.3 and 2.7. This isn't just a test of your hands, it's a test of your brain. And finally, you have got to be fluent in this vocabulary. If you can confidently explain how you used filtration to separate the excess insoluble base after the neutralization reaction before using crystallization to get your soluble salt, well, that's pretty much a perfect top-tier answer right there. And I'll leave you with this final thought. Our whole method, everything we just did, depended on the base being insoluble, right? That's what let us add it in excess and then just filter it out. But what if your base was soluble, like an alkali? You couldn't just add extra and filter it away, could you? It'd just be dissolved in the water. So how would you know the exact moment to stop adding it? How would you know when the acid was perfectly neutralized? That's a whole different challenge, isn't it?